which is good. That helps us move a little bit faster today. So the goals for, oh, please go ahead.
Sorry.
No, not at all.
For some reason, oh, actually, never mind. Sorry.
No worries. Everybody's happy and relaxed, right? Uh-oh, the back half of the room is not looking up. So our goals for this class and next class are to be able to, most simply, it's most basic, look at a pedigree, identify the inheritance pattern. This is dominant, recessive, X-linked, autosomal, things you might have seen in classes before. We're going to go into a little bit more detail. One of those details we're going to go into is a topic called incomplete penetrance. This basically means this isn't working exactly the way I would predict. So this is a fancy term that geneticists use for, hey, something's not quite 100% agreeing with the transmission pattern. Your response, say it with me, incomplete penetrance. Everybody? Incomplete penetrance. Anytime that you see a pedigree and you think you know what the transmission pattern is, but there's one individual in the pedigree that screws up the pattern, that's incomplete penetrance. That's your out. Another thing we can do with pedigree analysis, now that you understand Mendelian genetics, maybe you did before, when you look at a pedigree, we can also do some maybe genetic counseling. Tell two individuals, what's the probability that you're going to have a kid that has this, that inherits this genetic disorder? And finally, we'll talk about how geneticists actually identify which parts of which chromosomes cause genetic disorders. That's this bottom bullet point, genetic mapping. I threw this slide in just in case we had to talk about pedigree symbols at all, but your quizzes look like we can skip right past that. Does anybody have any questions about any of these symbols or actually drawing pedigrees? Otherwise, we're going to get right into looking at some pedigrees. Why is it the males that have to be the squares?
Does affected always mean genes, or is it any gene or like any trait that's passed down?
So affected, it has a sort of a negative connotation. I'm affected because I'm a male. You could call it, male is a phenotype. So basically affected means there's a phenotype that you're talking about. It doesn't have to have a negative connotation. Usually medical geneticists are talking about diseases. And so oftentimes when pedigrees are used for pedigree analysis, it's because a family has come forward to a physician and said, hey, we all suffer from this disease. And that's when the symbols get colored in, and a geneticist or a genetic counselor tries to figure out the transmission pattern. But we could map any trait onto a pedigree. Hair color, skin color, male versus female, which is already mapped by the shapes, right? Squares are males, circles are females. That's a trait. So we could fill in all the males. You're afflicted with having a Y chromosome. Yeah. So the first thing we should talk about is the most simple part of pedigree analysis. How do you identify a dominant versus a recessive pedigree? So here's the same pedigree. I'm going to fill this top one in, just with who's afflicted. This could be a disease. It could just be some other trait. So these individuals all have this disorder. Here's a family that's come forward to a genetic counselor and said, we've got this. These individuals have the disease. The maternal or paternal grandparent, right? So oftentimes in every generation we'll use maybe Roman numerals to label the generation numbers. So this might be individual 1A and 1B, grandparent, male grandparents, individual 1A in this case. So paternal grandparent has a disorder and two of his grandkids, one male, one female. Is this recessive or is it dominant? Well, why didn't Grandpa pass this disease on to his son. How do you explain that? Pardon? So if the son's a heterozygote, so we're going to work on some, one of you said, do you think this trait's recessive? And this is how every pedigree analysis starts. You make a prediction, you look at the pedigree, you make a prediction, you just ask, are all of the data in this pedigree consistent with that prediction? Is there any way that these patterns don't agree, don't support that this is recessive? So if this is recessive, then you're saying that individual 2A, the son, is heterozygous. How do we fill that in on the pedigree? Half filled in, right? This individual, if this is a recessive disorder, that individual would be a carrier. One of the two alleles of that gene is the mutant version that normally causes the disease in the recessive state, but because this individual is half unfilled in, it's got one wild type allele, the recessive phenotype doesn't show up. So this person has the possibility to pass on the trait, but doesn't actually exhibit the trait. Questions? I think I'm with you so far. So if this is what the parents look like, generation 2, father A is a carrier. Let's say mom is a carrier too. You wouldn't know this just by looking at her. Someone would have to tell you, or you would have to genotype her and find out if she was a carrier. But let's assume that the mom in generation 2 is a carrier also. Yeah?
If like the mom wasn't a carrier, then how could you get the thing? So you just have to.
Right. In this
genotype, she has to be a.
in this pedigree, if right, the assumption is this is recessive. That's what we're working on. How would you get any affected kids if there was only one allele present in the parents, one mutant allele? That is, in other words, if mom were wild type, would any of the kids show the phenotype if this is recessive? Yeah, you agree? No, they wouldn't. Yeah, no. If this is a recessive trait, let's say individual 2A is mutant asterisk over plus wild type. So if those are the genotypes of the mom and the dad in generation 2, what are the genotypes of all their kids going to be? Do the Punnett square if you want. Two options.
Homozygous dominant or heterozygous?
So you either inherit 2 pluses, you're homozygous, or you would inherit the mutation with a plus. That individual would be a carrier, they wouldn't have the phenotype. But in this pedigree, we have 2 individuals that are affected. Their genotypes have to be, if this is recessive, they have to have both copies of that gene be mutant. So since they're affected, what does the mom's genotype have to be? But mom's not homozygous recessive because she doesn't have the trait. She's half filled in.
Oh.
Right? So the mom's the carrier also. So the only way to get these individuals that have 2 copies of the mutation is one mutation had to come from each parent. So each parent, if neither parent exhibits the phenotype, which is true, these aren't fully filled in circles, they're just half filled in. So this is why recessive pedigrees or recessive traits skip a generation. Usually, not always, most of the time. So neither of those parents in generation 2 exhibit the phenotype, but they're able to pass it on to some of their kids. The kids that happen to, in bad luck, inherit the mutant version of the same chromosome from both of their parents. And what's the probability that dad in generation 2 happens to fall in love with and marry mom in generation 2, and they both happen to be carriers. They're pretty likely or pretty unlikely? It depends on the gene, depends on how frequent that mutation is in the population. This is why inbreeding is bad. This is why most human populations avoid inbreeding, because if you marry people that are related to you, you increase the probability that two of you happen to, by chance, have the same mutation because you're related to each other. So when individuals, the most often time you see this when two carriers marry and have kids and all of a sudden a phenotype shows up in their kids, those two individuals in generation 2 are often cousins, second cousins, third cousins, something like that. So recessive pedigree skips a generation. Recessive trait skips a generation in a pedigree, usually. Questions about recessive traits. That's the main way we identify them. You see it in one generation, it's absent in the next generation, it shows up again in the third generation. So by contrast, what is this going to look like if we do a dominant trait in the bottom? So is generation 1's son going to have this disease? Depends on, so if this is dominant and mom's not affected, what's mom's genotype? We can already figure this. This is why I love genetics. It's a big puzzle. We already know mom's genotype. If this is dominant, what's mom's genotype? Homozygous wild type, yeah, recessive. Now, what's dad's genotype? This we don't know. He either has one copy or two copies of the dominant mutation, so we don't know if he's homozygous or heterozygous. So is the son going to be afflicted with this disease or not? What's the probability that the son in generation 2 is affected? 40%, 40, going once, going twice, I heard 50%. Right, so the most likely default assumption, most likely is that dad's a carrier. That is, he's heterozygous. He's affected, so he's not a carrier, but asterisk over plus, right? Dominant mutation, one copy gives you the phenotype, the disease. If this is the case, then his son has a 50-50 chance of either inheriting the mutation or the wild type allele from his dad. So let's say in this case that that son actually does inherit the trait from his father. So he was in the unlucky 50%. And let's say he's married a wild type woman, or not married, I don't know. You make up the story if you want to. What happened in Vegas stayed in Vegas. They happened to have 8 kids while they were there.
It's always the prize, just 8 children, you know?
Hit the jackpot. What's going to happen in the third generation if this is a dominant disease and dad's got it? How many of the 8 kids should we expect to see filled in? 50%, right? Every one of them has a 50-50 chance of inheriting dad's good allele or his mutant allele. So I'll just fill in every other kid, I guess, just for fun. We don't know which of these kids will have it, which won't, but on average, if you look at a big family, half the kids should have the dominant disorder, half don't. So this is why the dominant diseases tend, this is not an absolute rule, this is a pattern, tend not to skip generations. You tend to see individuals in every generation have the disease.
Dr. Ross?
Yeah.
You're saying this for diseases, I'm guessing this also works for traits?
Exactly. So yes, when I say disease, you could think phenotype, you could think trait. Absolutely. Okay. So here's something else we can do with pedigrees. And this is an important nomenclature slide. This isn't in the textbook, so this is important for understanding what we're going to do for this class and for next class. Here I've just diagrammed a pair of chromosomes, as I often do, as vertical sticks. But I'm using a new form of nomenclature starting today. So here what I'm trying to show you is that we've got a couple of dinucleotide repeats, in this case a CG. The chromosome on the left has 1, 2, 3 CGs, which we've talked about before. That's the nomenclature. There are 3 copies of CG in a row on that haplotype, and the other chromosome has 2 copies, so it's a CG2 allele. It's the number that we're going to focus on today. The chromosome on the left has 3 copies. The chromosome on the right has 2 copies. And this is how human geneticists tend to name alleles. So let's assume that this is chromosome 1. Every gene or every locus we look at along the chromosome as we go from top to bottom will just be given, let's say, a number. So we've got chromosome 1. Both of those are on chromosome 1. We've got 4 copies of T in a row, reading from 5' to 3'. So we've got 4 copies of T here on that chromosome. That distinguishes it from the other chromosome that this individual has, which has only 3. We've got T4 on the left, T3 on the right. So these are both chromosome 1. We're going to call the top locus, locus A. The second one we'll just call locus B. So we'll just go down from the top to the bottom. Chromosome 1, gene A, chromosome 1, gene B, chromosome 1, gene C, and so on, from the top to the bottom of the chromosome. And then the third thing that's going to be making up these allele names is just the number of repeats. So the individual here on the left, this is 1A3. Chromosome 1, A is the first gene or locus we're looking at, and 3 is the number of repeats in that dinucleotide, trinucleotide, whatever it happens to be. So what would be the name of the haplotype on the right here? What do we call this version of that allele? Which chromosome? Still chromosome 1. Which gene or which locus? It's the same region, so this is 1A. This is just a different version of 1A. Say it louder. 2. Right, this is the version of the chromosome that has 2 CG repeats. So we're just building up a nomenclature for how we refer to different versions of different regions of different chromosomes. Chromosome number first, then the gene or locus letter or designation, and then some description of the allele. So let's fill in the bottom 2. Got chromosome 1, locus B. Chromosome on the left is how many T's? 4. This one's 1B. 3. You're going to see this nomenclature show up again. That's what it means when we talk about alleles that have these particular nomenclatures. Chromosome number, locus, and then which version of the allele is the last, the third character. 3, 4, 2, 17, 25, whatever. Questions or concerns? So here we can do something with pedigrees that we normally can do. We've got the parents' genotypes up here. I zoomed out a bit too far. So we could fill in what the kids' genotypes are likely to be. So help me, this is another puzzle. So we're trying to figure out what goes in these blanks. We've got partial genotypes of some of the kids. Can we figure out what goes here, for example? So kid 2A's got CG3 on one of their chromosomes, one of her chromosomes. Which parent did that have to come from? Which parent could CG3 have been inherited from? Which parents have that version of that gene? Both. Okay, so that doesn't help us. We don't know yet whether or not this CG3 came from the mom or came from the dad. So what are the possible alleles that would fit in there? If CG3 came from the mom, what are the options of alleles that came from the dad? It could be 3, or it could be dad's other allele. Right, so we don't know if this kid has 3 or 4. I shouldn't use a slash probably. It's not a genotype, right? That kid must have inherited one or the other versions from their dad. Yeah.
We know for sure he inherited the left side from the mom because there's a T4 on the bottom.
Ah. So if we've got T4 down here on the bottom, that had to come from which parent? It's unique to the mom. So what has to go here? Dad's version. Dad is homozygous, so this kid has to have inherited, we don't know which physical molecule. It doesn't matter. They're both identical. They're both T3. So we're unsure. We can't, this is where it's really important to be clear. There's not always a correct answer. There's not always an answer when you're doing pedigree analysis. We can't tell with the information we have what the genotype up here is. We can guess. We don't have any evidence to choose one or the other. It's either version 3 or version 4. What about this? This blank down here. Son 2B has T3. Which parent did that come from? Could have come from either. So since we can't rule that out, then we're back to the same issue. It's either T3 or 4 that comes from the other parent at this locus.
Couldn't you actually figure it out since the mom has CG2 and that's the only copy of CG2 that would have to be T3?
Okay, so this is a fabulous question. If mom has CG2 and T3 on the same chromosome, and this kid has CG2, must have gotten that from mom, right? Mom's the only person in the parental generation that carries that allele, the version 2. Why does that not help us figure out what's happening at the other locus? Crossing over. So we don't know if when mom made her gametes that there might have been a crossing over event up here that made CG2 linked to T4. So we really can't distinguish whether or not, at this point, whether or not, without taking the DNA from that son, 2B, we can't tell what his genotype is likely to be. So we've done as much as we can trying to figure out genotypes based on possible inheritance patterns. Why don't we, let's do something crazy and just pick one. For his son, 2B, do you want him to have received allele 3 or allele 4? This is as crazy as geneticists get. Help me out. 3. Okay, so we're just going to guess that that's allele T3. Okay. So now I'm going to give you a little bit more information. These individuals are all afflicted with some sort of phenotype. So 1B, 2B, and 2C all have, I don't know, you decide, some phenotype, freckles. Here's where the human geneticists and the genetic counselors come in. So we're trying to figure out what mutation or what allele causes this inheritance pattern. What's genetically common that we can see up here between all of the individuals that have this disorder and their genotypes? We're looking for correlation. So they have correlation between their genotype on a particular locus on a particular chromosome and their phenotype. They have a disease or they don't.
They don't have T4.
Pardon?
They don't have T4. The affected don't have T4.
So everybody who is unaffected, 1A and 2A, both have T4 at the bottom locus. The others, what's true about their T locus? They're all homozygous for T3. So there's a correlation. Everybody who's affected by this disorder is homozygous for T3. Yeah.
Also that, for the people that are affected, the children, both of them have a T3 and a CG3 that contribute to the disease, because also the parent that's afflicted also has a T3 and a CG3.
Okay, so the affected individuals also all have CG3s. Someone help us out. Why are we not making a big deal about that?
How do you know if it's just the T and not, you don't look at the CG3 or you can't look at both?
The unaffected one also has T4.
Right. So there's no correlation between whether or not you have CG3 and whether or not you have the disease. Because there's some unaffected individuals up here that have CG3. So that's why we don't pay attention to the CG3 in this case.
Could it be that the CG3 becomes active or something with the disease when it's found in that T3? Because then the other parent that's not affected, and they have this CG3, but
So-
it's not on the same chromosome.
So now we're talking about complex traits. Multiple genes controlling, and absolutely that could be the case. I'm trying to do simpler, but yes, you're absolutely right. Real human geneticists, when they're doing genetic counseling, have to worry about those sorts of things. You've got 23 chromosomes to worry about. Does the genotype at one point on one chromosome depend, or does the phenotype depend on a genotype here on this chromosome and your genotype over here on this chromosome as well? But simple rules first. all we're doing to try to identify simple bases of genetic disorders is just to look at correlation between genotype and phenotype. So who's got the disease? What do they share genetically to the exclusion of everybody else who doesn't have the disease? It's the simplest way to start. That doesn't mean that it's not complex, it's just how we begin.
If the unaffected was homozygous for CD3, would you say that would play a part or no?
If the unaffected were homozygous for CG3, which individual? Any individual?
Like if the first one was CD3 and it was T4, it had the T4, like would you say the CD3 and the T4 are causing it to be unaffected?
Right, so if there was also a perfect correlation at the top locus, then you would have to say that it might also be involved. Right. So we're just trying to get basic principles here because we're going to apply this to more complicated examples in just a minute. This is the most simple version. Look for correlation between genotype and phenotype. Question in the back? So all we're doing is we're looking for what's common, what's genetically common among the individuals that have a particular phenotype. Okay, right, correlation and causation are really critical for this, and we're going to start next class talking about correlation and causation. But I wanted to point out, thank you very much for mentioning this, that I'd skipped past this. Note that on this chromosome, there's a difference in the number of T's. There are two different alleles, T4 on the left and T3 on the right. What's right underneath those T4 and T3 alleles? What's the next nucleotide? On one chromosome, it's an AT base pair. On the other chromosome, it's CG. This is a single nucleotide polymorphism, or SNP, right? A single base pair change. So we're looking at inheritance patterns using 1b, a locus that we call 1b. It's on gene chromosome 1, it's locus B. But does it matter whether or not you have 4 copies of T or 3 copies of T? Is that causing the disease that's present in this pedigree that's just off the screen here? Or is it perhaps this single nucleotide difference between the chromosome on the left and the chromosome on the right? How would you describe the relationship between T4, the T4 allele, and having an A? Those alleles are on the same physical piece of DNA. How close together are they? Very. So T4, you fill in the blank. T4 is blank to A. We did this last chapter. There's a big test on, say it louder.
Linked.
They're linked. Right? Allele T4 is physically linked to having A. What's linked to T3? G. So if you see that somebody has inherited T4, do you know for sure that that causes a disease, that it doesn't actually happen to be the SNP? That if you inherit T4, you also inherit it at the same time in A. It's that one spot on the chromosome. Which is it that causes the disease? Is it the T4 or is it this single nucleotide polymorphism?
Is it okay we don't know?
We don't know. Yes, please. It's very much okay if you don't know. That's the point, is that all genetic mapping does is look for correlations. We see that an individual has T4.
Because wouldn't having like T4, 4 Ts, wouldn't that also decrease like how cohesive the DNA is?
So there's all sorts of things that we could, there are all sorts of stories we could make up about how this mutation might affect an individual. The simple point is that we use these genetic markers, we call them, these loci, 1A, 1B, and so forth, just to track the inheritance of segments of DNA from one generation to the next. That inheritance pattern doesn't necessarily mean that those specific pieces of the chromosome that we're looking at generation after generation are what caused the disease. It just means something linked to that locus. Some gene close by is likely causing the disease. So it's not, I just want to make that really explicitly clear, that it's not the specific pieces of DNA we're looking at generation to generation, the specific genes or loci that are causing, they're just correlated with. So we could also do the reverse. We can get genotypes of all of the kids. This is more likely in human genetics. It's more likely that we have DNA from the current generation, but it's harder and harder to get DNA from people that are older or perhaps dead. So here we have full genotypes for a set of kids. We're trying to figure out the genotypes of their parents. So go ahead and work on this in pairs or groups, small groups. I'll give you like 2 minutes. Try to figure out what goes in those 2 blanks in the genotypes in the first generation. So here, let me tell you this. Let me say this individual is affected. And this one too. There you go. Okay. So here's a bigger pedigree. What are the correlations between who's affected and disease state, just in these 2 individuals? What are the things that could possibly be causing 1B to have a genetic disorder and 1A not to? What's unique to individual 1B? Okay. 1B7. 2A7. Okay. So let's assume that this is brachydactyly, just to put a name you might be familiar with, right? The short fingers. So those are the possible associations just in this one generation. We're just looking at 2 individuals. So far, you've already made a prediction about where you're going to look in subsequent generations to ask whether, which of these regions, we've got 2 chromosomes, which chromosome numbers are we looking at here? If we read the allele designations, chromosome 1's on the top, then chromosome 2. Right, so we've got gene on chromosome 1 named A, locus 1B, locus 1C, locus 2A, locus 2B. Okay, so if you move forward a slide, you can look at the full pedigree with everybody's genotypes. Your prediction is that either 1A4, 1B7, or 2A7 causes this disease. Holy crap, look at all that information. Where do you start? What can we ignore in this picture to do this analysis? What region of which chromosome controls this trait?
You can ignore 2E, 2F.
You ignore 2E, 2F. Yeah, don't pay attention right now to anybody who's unaffected. Just look for what's common between all the filled in symbols. So we started off saying it was either 1A4 or 1B7, or possibly 2A7. So which of those are common to everybody that has this? So every individual that has this disease has inherited from grandpa in this bottom generation. 1A4 and 1B7. Is there anybody who's unaffected? Now we can look at the empty squares. Is there anybody who's unaffected that has 1A4 or 1B7? This is an important double checking step. Nobody else, I think, has 1A4 or 1B7. So it looks like there's a perfect correlation between your inheritance of this region of chromosome 1 and whether or not you get the disease. Does this look like dominant or recessive? It's dominant. It's in every generation. And it only takes being heterozygous. You don't have to have 2 copies of 1A4 or 1B7. 1 copy, you're correlated with the disease. Now, what about chromosome 2? So here we're getting back at one of your original questions. What if there are 2 genes on 2 separate chromosomes that together control this trait? If we look at 2A7, is there a perfect correlation between who's got 2A7 and who's got the disease? Well, here's a diseased individual that has the disease and does not have any 2A7. So we rule that out as being a cause of this genetic disease. So in this case, it looks like there's a single locus, a gene somewhere in that region of the chromosome that's a dominant allele that causes this disorder. I have 2 requests for you. I know I'm a minute late. That's it for content. I have 2 requests for you for next class, though, so hang on just a second. Here's a real pedigree. Oh, I know. From published literature way decades ago. This is a family from Central America who has autosomal deafness, so heritable deafness. This is laid out exactly the same way that we just looked at in the cartoon pedigree on the previous slide. So there's some affected individuals, the chromosomes are arranged top to bottom. Your job for next class, there are 2 jobs. One is to figure out, circle, which region of the chromosome is correlated with deafness in this pedigree, which region of this chromosome. And then I'm also going to open up a Google, so that's going to be a Google exercise. So the PDF is going to go out to you, just circle on the PDF which part of the chromosome you think causes deafness. I'd also like you to draw your own pedigree and send it to me. You don't have to put your information on it, but I want to make sure everybody understands pedigree drawing. Next time, I'm going to share my pedigree with you, and we're going to talk about one of my favorite genetic disorders when we start talking about sex-linked traits, X-linked, Y-linked, and so forth. And thank you very much for listening. Go forth, and it's office hours if anybody wants to come talk. Yes?
Does that number at the end mean anything? Like, I know the 1, the first number means the chromosome, the A, and the A.
So in the real pedigree, in the deafness pedigree, yeah, so the 7 is, going back to the original picture, the original diagram of nomenclature. In this case, 7 is often the number of repeats of dinucleotide and trinucleotide. Okay. So you could have individuals that have 1, 2, 3, 4, 5, 6, 7, 8, whatever. But often, a single pedigree, there will only be a few different versions. You might see like 1A7, 1A12, 1A21. So it's not necessarily that there is a 1, 2, 3, 4 forever. So, do you want your reading assignments to show up in Blackboard?
Well,